Hello everyone. Today we'll be solving Cambridge International AS and A level biology paper 1 multiple choice February-March 2021 variant 12. The photograph shows onion cells. We can see Y, a single onion cell is basically measured. Y is equals to 14 centimeter. The actual length of the onion cell is 350 micrometer. What is the magnification of the photomicrograph? So we know magnification is equals to image divided by actual. The image in this case is 14 centimeter. So to convert it into millimeter, multiply it by 10. To convert it into micrometer, we will multiply it by 1,000. And then we will divide it with 350, which is the actual length of the cell. After dividing, our answer comes 400. So D is the correct answer. Which row correctly shows the presence of cell structure in typical plant cells and in typical prokaryotic cells? In typical plant cells, we do not have centrioles. Typical prokaryotic cells and typical plant cells both have 70S ribosome because plant cells contain mitochondria. And plant cells contain mitochondria and chloroplast, which are both, it contains mitochondria and chloroplast, which are both early bacteria. They are in the eukaryotic cell in a endosymbiosis. So we know that typical prokaryotic cells does not contain any membrane-bound organelles, and cellulose cell walls is not contained within typical prokaryotic cell. So all of the options are wrong. Option number 2, B. Which letter identifies cell structure where semi-conservative replication DNA occurs? Semi-conservative replication of DNA will occur in chloroplast because they have their own DNA, in mitochondria because they have their own DNA, and also in nuclei because they have, in nuclei we have the DNA. So option A will be correct for all of them. Which parts of a cell contains ribosome? We know that chloroplast, it contains its own genetic material and also contains its own ribosome. Mitochondria contains its own ribosome. Golgi body does not contain ribosome, and cytoplasm contains ribosome. So option 1, 2, and 4 will be correct. So option C is correct. Which range of cell diameters is typical for prokaryotic cells? We know that a typical prokaryotic cell can be 1 micrometer to 5 micrometer. So in this case, 1 nanometer is too small, so that's wrong. 1 into 10 to the power 3 nanometer. This represents 1 micrometer. So 1 to 5 micrometer. Option B will be the correct answer. Which row shows a combination of molecules that could be found together in a single isolated virus? So in a single isolated virus, there will be no carbohydrate. There could be DNA. Phospholipids, there is no chance. Lipids, no. Proteins, yes, there should be a protein coat. And RNA, well, may not be present. So option D is the correct answer. A student carried out 4 biochemical tests on a sample of food. Test. Emulsion test, it goes cloudy. Emulsion test going cloudy is an indication of presence of fat. Benedict test, it went yellow, so there is reducing sugar. Biuret goes purple, so there is a protein present. Iodine, yellow, no starch. What conclusions is supported by the result? Fat is not present, no. Glucose is present, well, cannot confirm that. Protein is present, yes, we can confirm that. Starch is present, no, starch is not present. So C is the correct answer. Question number 8. Which diagram represents a molecule of alpha glucose? A typical molecule of alpha glucose will have the first carbon position OH at the bottom. Which diagram represents a molecule of alpha glucose? So we know that alpha glucose, in the first carbon, the OH will be at the bottom. And in the fourth carbon, the OH will be at the bottom. And in the second carbon, the OH will be at the bottom. In the third carbon, the OH will be inside. So A is the correct answer. Let's see why other options are not correct. We can see both of these OH are inside, which is not a typical of alpha glucose. Alpha glucose does not have the first OH at the top, and neither have the fourth OH at the top. So option B, C, D are obviously wrong. Which statements about glycosidic bonds are correct? Condensation between glucose and fructose uses water and forms sucrose. Condensation reaction does not use water. Option 1 is wrong. We can see if option 1 is wrong, then option A, B is already gone. So we have 2 and 3 and 2 and 4. So option 2 will always be correct. So we can go for option 3. The hydrolysis of amylopectin uses water to break glycosidic bonds and releases alpha glucose. Yes, amylopectin is made of alpha glucose. Option 3 is correct. So C should be correct. Amylose contains 1,6 glycosidic bond. Amylose only contains 1,4 glycosidic bond. So option 4 is wrong. So 9C. Our triglyceride has obviously it has ester bonds. Okay, so option number 1 is ester. Bonds formed by joining fatty acid to, so fatty acids are joined to glycerol. It is not joined to glycine. So option C is the correct answer. Question number 11. The diagram shows a phospholipid molecule divided into 3 regions, X, Y, Z, and R. In region X represents a range of possible chemical groups. All right. So we can see the X mainly shows, this is a phospholipid. The X shows the phosphate head. The phosphate head. There must be, something else with it as well. Z is a unsaturated hydrocarbon. Unsaturated hydrocarbon prevents close packing. And Y is a saturated hydrocarbon. The reason XYZ affects the properties of cell surface membrane in different ways. Which row shows the effect of each region on the properties of a cell surface membrane? Increases permeability of the hydrophobic region. So hydrophobic region, the permeability can be increased by saturated hydrocarbon. All right, because it is, twisted, it increases the amount of space it needs. So Z does it. So repels polar molecules. Y and Z both repels polar molecules. All right, attracts water molecules. All right, X, the region X attract water molecules, which is the phospholipid. So D is the correct answer. 11D. Question number 12. Which statement about collagen is correct? Collagen. Tissues containing collagen fibers cannot stretch. Well, that is not true. Collagen is flexible. Each polypeptide chain forms an alpha helix. Collagen forms helices, but it is not alpha helix. 3 helices are held together by hydrogen bonds. Yes, 3 helices are held together by hydrogen bond in collagen. So option C is the correct answer. Let's see option D. 3 polypeptide chains forms a single collagen fiber. No, that is obviously not correct. Catechol, question number 13. Catechol is chemical found in a number of fruits, catechol can be oxidized to a quinone by the enzyme catechol oxidase. Catechol oxidase is inhibited by para-hydroxybenzoic acid, PHBA. This, which is structurally similar to catechol. In the presence of PHBA, the Michaelis-Menten constant Km for the catechol oxidase increases. All right. How does PHBA inhibit catechol oxidase? So when we go through this particular information, so we can see, we find out the information that catechol is a chemical that is found in a number of fruits. It is oxidized to a quinone, and the oxidizing agent is basically an enzyme which name is catechol oxidase. All right. Now we see that catechol oxidase can be inhibited by PHBA, para-hydroxybenzoic acid, which is a naturally similar to catechol. When it says naturally similar to catechol, we get the understanding that this is a competitive inhibitor. Competitive inhibitor. All right. So now the next point that is given, in the presence of PHBA, the Michaelis-Menten constant Km for catechol oxidase increases, which is also a telltale indication. If the Km value increases, meaning that enzyme affinity towards towards the substrate has decreased, which is a telltale indication of competitive inhibitor. So PHBA is a competitive inhibitor. How does PHBA inhibit catechol oxidase? PHBA and catechol can both bind to the enzyme, but not at the same time. Inhibit catechol oxidase. All right. So, I mean, they can both bind to the enzyme, that is correct, but not at the same time. All right, which is also correct. I mean, but then how does PHBA catechol oxidase inhibits catechol oxidase? All right, PHB is a non-competitive inhibitor. We can see option 2 is definitely wrong. If option 2 is wrong, then definitely it will be either option 1 or option 2, because PHBA is not a non-competitive inhibitor. PHB decrease. Okay, the third point says PHBA decreases the Vmax of the reaction. So we know that a non-competitive inhibitor does not decrease the Vmax of the reaction. So this is also wrong. So only number 1 is correct. B is the correct answer. All right. So PHBA and catechol can both bind to the enzyme, but not at the same time. Question number 14. Which statements about the fluid mosaic model of a membrane are correct? The movement of phospholipids in the membrane is reduced if the fatty acid tails are saturated. All right, that is true, because if the fatty acid tails are saturated, they are going to closely pack with each other, and thus phospholipid bilayer is very less likely to move. All right, the glycoproteins in the outer layer of the membrane can move. That is definitely correct. Channel proteins are fixed in position. Well, channel proteins are not fixed in position. They can move around as well with the phospholipid bilayer. So 1 and 2 is correct. Option B is correct. The diagram 1 and diagram 2 shows how the transverse section through a leaf changes when the leaf is moved from a solution X to solution Y. Guys, this kind of questions can be very tricky. We can see in this particular diagram, the leaf is curling up. The leaf curling up is a telltale indication that the solution is very concentrated. So we can say this is a solution with low water potential, or this is a concentrated solution, a concentrated solution. We can understand that, obviously. And this is a dilute solution. Because we can see the solution Y allows the leaf to be turgid. So this is a dilute solution. All right. Now that part being clear, obviously the net movement of water will be towards the solution. The net movement of water from solution Y will be towards the leaf. Which row describes the water potential of the leaf cells and surrounding solutions in diagram 2 compared with diagram 1? All right, so which row describes the water potential of the leaf cells and the surrounding solution in diagram 2 compared to diagram 1? Okay, let's see. First option, water potential of the leaf cells in diagram 2 compared to water potential of leaf cells in diagram 1. All right, the first option says less negative. All right, in order to understand that, we can do the comparison by ourselves. So we have to look at the water potential of the leaf in diagram 2, all right, compared to water potential leaf in diagram 1. Because the diagram 2 is gaining water, so the water potential will be, the high water potential, obviously. High water potential. All right, and this one is losing water, so it's not gonna lose the solute, so it's gonna be low water potential. So since low water potential means more negative, so high water potential means more positive. So yeah, all right, it will be less negative then. So less negative. All right, so less negative, both correct. Water potential of the solution Y compared with water potential water potential of solution X. So yeah, the solution Y is dilute, so it will be less negative. All right, so less negative, both parties satisfies A. 15A. Which feature must always be present for water to move between two solutions by osmosis? All right. Okay, so of course, there is a need for carrier proteins if water were to move between two, between two solutions by osmosis. All right. Which feature must always be present for water to move between two solutions by osmosis? Well, we do not need carrier proteins to move water. We do not also need cell surface membrane always to move water between two solutions between osmosis, but we definitely need a selectively permeable membrane. The reason why we say that we do not need a cell membrane, because you see, osmosis can also be done in a Weisking tubing. Now, a Weisking tubing obviously does not have cell surface membrane, but yet it will allow osmosis to occur. Okay, water potential gradient must be also present. Yes, definitely water potential must be, gradient must be present in order for the water to move from a region of high water potential to low water potential. So option D is the correct answer. 17. When living pancreatic cells were placed in solutions of red stain called neutral red, the cytoplasm became red. The cells were then removed from the solution of neutral red. The red stain in the cytoplasm moved into vesicles. All right, so there we can see it's an example of diffusion, and which were then exported from the cell, eventually leaving the cell colorless. Now we can see exocytosis because they were put into vesicles. The vesicle, emptied them out. Which transport mechanism could explain how the red stain entered and left the cells? So definitely there were diffusion. All right, there were diffusion. So active transport and facilitated diffusion does not help here. There were diffusion, there were exocytosis. So B is the correct answer. Question number 18. DNA molecules in eukaryotic cells must be packed compactly because their combined length is much greater than the diameter of the nucleus. Which structure are required during the process of packaging DNA molecules compactly? Now we know histone proteins are needed. to compact DNA molecules, very neatly. So yes, telomeres, no. Chromosomes, no. Chromatids, no. Question number 19. Which statement about mitosis are correct? Mitosis results in genetically identical cells. Yes. Mitosis is involved in asexual reproduction. Yes. Mitosis occurs immediately after cytokinesis. No. Mitosis can be used to repair damaged cells. No. All right. So option 1 and 2 correct. Definitely C. Question number 20. A human chromosome is made up of different parts. Some parts of a typical human chromosome are more numerous than others. Which parts are listed in order from least numerous to most numerous in a human white blood cell? All right. So you see, the question says, all right, which parts that are listed most numerous in a human white blood cell? So least numerous to most numerous. All right, one thing that we can tell for sure, all right, centromere is very limited because each chromosome has only one centromere. Chromatids will be double the number of centromere in that regard, and followed by histone protein. So option A looks like the perfect answer. Let's see the other options. DNA molecule, telomeres. There are more telomeres than DNA molecules. All right, histone proteins, and then telomere. Well, telomere, histone proteins are more compared to telomere. Telomere, centromere, and chromatid. So there are more telomere than centromere because it says from least numerous to most numerous. So option A is correct answer. A piece of double-stranded DNA containing 12 into 10 to the power of 3 nucleotides is transcribed and translated to produce a single polypeptide. What is the maximum number of amino acid in this polypeptide? Okay, so the question says, a piece of double-stranded DNA containing 12 into 10 to the power of 3 nucleotides. All right, it is transcribed, all right, translated. Now you see, DNA is double-stranded. Since the DNA is double-stranded, if there is 12 into 10 to the power of 3 nucleotides, so 10 to the power of 3, that means that is 12,000. Then we must divide it by 2 because it's double-stranded. So the answer will be 6,000. And we know that each amino acid requires 3 DNA bases. So divided by 3, the number comes to 2,000. So 2,000 is equals to 2 into 10 to the power of 3. So option C is the correct answer. All right, moving to question number 22. Which statements about complementary base pairings are correct? Cytosine forms 2 hydrogen bonds with guanine. Cytosine forms 3 hydrogen bonds with guanine, so option 1 is wrong. If option 1 is wrong, fortunately, this question is very easy. Then option 2, 3, 4 should be correct, and D should be correct. All right, so question number 22, the answer is D. All right, so let's check the other options. Purines and pyrimidines are different sizes. Yes, definitely they are. Adenine forms the same number of hydrogen bonds with thymine as it does with uracil. Yes, it does. 2 hydrogen bonds in both cases. The base pairs in a molecule of DNA are equal length and equal width. Base pairs, 2 base pairs together in a molecule of DNA, they are always equal length. So yes, point number 4 is correct. What are the products when a DNA molecule replicates? What are the products when a DNA molecule replicates? Okay, so, if a DNA molecule replicates, we're going to get 2 DNA molecules, that's for sure. So 2 molecules of DNA, 2 molecules of DNA are correct, whereas 2 strands of DNA are wrong. Now, however, each made up of sequence of paired bases, well, They're not made up of bases, they're made up of nucleotides. So each will be made up of sequence of paired nucleotides. So option B is the correct answer. Going to question number 24. Which descriptions apply to phloem sieve tube and to xylem vessel elements? There is no cytoplasm. No, there is cytoplasm in the phloem sieve tube. All right. There is no end walls. No, there is end walls in phloem sieve tube. There is no nucleus. Yes, both of them does not have nucleus. So option 3 is the correct answer. Option D is the correct answer. 24D. Some of the parts of a plant root involved in water transport are listed. All right. Some of the parts of a plant root involved in water transport. Okay. All right. We can see Casparian strips, cell walls of the cortex, endodermal cells, epidermal cells, xylem vessels. All right. So which sequence shows part of a pathway through which water can move across a root? All right. Great question, actually. All right. So yes, of course, our main goal will be to go towards xylem. So point number 5 must be included. Xylem vessel must be included. So option C and D are both wrong. All right. Then option A and B both starts with 2. So cell walls of cortex cells. All right. After the cell walls of cortex cells, now, option number 1, it says Casparian strip. All right. And option number 3, Casparian strip, definitely not. Cell walls of cortex cells. Casparian strip is impermeable to water. It is impermeable to water. So water. So definitely it won't accept the water to pass through there. So after that, it must enter the endodermal cells. So option A is the correct answer. 2 to 3, then to 5. Okay. All right, moving to question number 26. What is an example of a mass flow system in plant? Good question. All right. The loading of amino acid from the surrounding tissue into the companion cell. Well, that's not a mass flow. All right. It is one type of, active transport, but it's done indirectly, definitely. The movement of sap through the phloem sieve tube in stem. Yes, definitely. Sap moving through the phloem sieve tube in the stem would definitely be mass flow. The movement of water across the root along the symplastic pathway, well, that is not mass flow. All right. The movement of water vapor from internal spaces in the leaf out through the stomata. Well, that is not mass flow as well. So option B is the correct answer. The diameter, question number 27. The diameter of a tree trunk usually decreases slightly during the day. That's because of the transpiration pool, which changes in environmental factor during the day could cause the diameter to decrease even more. All right. So increased humidity, definitely not. If there is increased humidity, then less transpiration will occur. Increased temperature, yes. Increased light intensity, yes. So option A will be the correct answer. 27A. Which statement about the role of co-transporter proteins in the movement of sucrose is correct? Co-transporter protein pumps hydrogen. No, definitely not. This is a big no. All right, co-transporter protein does not pump hydrogen. Hydrogen ion diffuse through co-transporter proteins with sucrose into companion cells. Yes, that is a function of a co-transporter. It co-transport H plus ion with the sucrose. Plasmodesmata move sucrose into phloem sieve tube via co-transporter. So plasmodesmata moves sucrose, all right, into phloem sieve tube via co-transporter protein. Plasmodesmata does not require a co-transporter. It's a direct cell membrane to membrane connection. Sucrose is moved through co-transporter protein by active transport. Well, it's not an active process. All right, so B is the correct answer. 28. Which row correctly describes the heart during atrial systole? During atrial systole, the walls of ventricle must be relaxed. All right, if it were to be contracted, then Atrial systole does not have enough power to push the blood into the ventricle, definitely. Atrioventricular valve must be open and semilunar valve must be closed. So option number D is the correct answer. Moving to question number 30. The diagram shows a capillary, some tissue cells, and lymph vessels. We can see the capillary. We can see some materials coming from the red blood cells directly into the tissue cells. So definitely could be diffusion of some kind of substances, specifically oxygen in this case. So option number 1, which row correctly, the question says which row correctly identifies arrows 1, 2, 3, 4. All right, so arrow number 1, definitely diffusion of oxygen. Diffusion of oxygen, both looks correct. Arrow number 2, flow of water and small solutes. All right, flow of water and small solutes are correct. Flow of water and plasma proteins. You see, from capillary, plasma proteins cannot leak out. All right, in typical condition, it won't leak out. So option number D is wrong. Now, for option number 3, diffusion of carbon dioxide, definitely it could be diffusion of carbon dioxide from the respiring cells into the plasma. And 4, flow of tissue fluid. Yes, 4 is flow of tissue fluid. So option C is the correct one. 30C. The table shows the result used to draw an oxygen dissociation curve for adult hemoglobin. Partial pressure of oxygen. All right, 1, and we got 0.11. Partial pressure of 7, we got 1.15. Volume of oxygen per gram of hemoglobin. All right, so 1.15. Okay. What is the percentage increase in the volume of oxygen per gram of hemoglobin when the partial pressure of oxygen increases from 3 kPa to 5 kPa? So if we look into the information, at 3 kPa, we were at 0.58. And 5 kPa, we were at 0.96. So 0.96 minus 0.58 divided by 0.58 into 100% should give us our answer. All right, so that equates up to 65.5%. So option number D is the correct answer. The graph shows the oxygen dissociation curves for adult hemoglobin at 2 carbon dioxide concentration. All right, we can see it too. So obviously the bottom one is at, all right, high partial pressure of CO2. All right, and this one is at low partial pressure of CO2. All right, now which letters show the measurements taken in the capillaries of the lungs and in the capillaries of active muscle? So in active muscle, definitely it's going to be R. Active muscle will be R because there is low partial pressure. There will be high partial pressure of carbon dioxide in active muscle. So R is the correct answer for active muscle. All right, and in the lungs there will be high partial pressure, low partial pressure of carbon dioxide because air contains less carbon dioxide. So that will be P. All right, R, and in the lungs it will be P. So A is the correct answer. 32A. Which substances can combine the adult hemoglobin and reduce the oxygen carrying capacity of the blood? All right, yes, we know that carbon dioxide can, bind with hemoglobin and produce carboxyhemoglobin. So definitely oxygen is unloaded after that. So yes, carbon monoxide definitely binds with hemoglobin and decreases oxygen carrying capacity. Hydrogen carbonate ions does not bind with hemoglobin. So hydrogen ions does bind with hemoglobin and cause the hemoglobin to lose oxygen. So option number 4 also is correct. 1, 2, and 4, option B is correct. How many times must a molecule of carbon monoxide pass through a cell surface membrane as it diffuses from an airspace in the alveolus through a cell in the capillary wall into a red blood cell? Tricky question. You see, if we were to draw an alveolus and the blood vessel that is passing right next to it, okay, you see the capillary in the blood vessel will contain two cell minimum, one cell thick, but there will be two cell membranes. So if a oxygen were to diffuse directly from there into a red blood cell, so it must go through two, three, four, five cell membrane before it reaches the hemoglobin to a red blood cell. So definitely, if it wants to go through a red blood cell, it must go into a red blood cell, it must pass the outer cell membrane of the red blood cell. 34 answer is C. Question number 35. The heart rate of a person at rest was recorded at intervals over 2 15-minute period. During the first 5 minutes of the second 15-minute period, the person smoked a cigarette. Okay. All right, we can see after smoking the cigarette, the person has a, pretty much high heart rate. The graph shows the results recorded during the 2 15 minutes period. What can be concluded from the graph? Carbon monoxide affects the heart rate. Carbon monoxide affects the heart rate. Well, we can't say that for sure, that carbon monoxide is the effect, the main reason for the heart rate to increase. That we can't tell for sure. The next point, it says carbon monoxide binds to hemoglobin, increasing the flow of blood. All right. Well, carbon monoxide does not increase the flow of blood. Nicotine increases the heart rate, so it's a stimulant. It doesn't prove that the smoke contains nicotine. So can confirm that. Heart rate increases as a result of smoking. Yes, we can definitely confirm that, because the person did not have any exercise or anything. It just the person smoked cigarette and the heart rate increased. So definitely smoking increases heart rate. Question number 35, option D. Emphysema is a type of chronic obstructive pulmonary disease. A person with emphysema has difficulties in breathing out. Why does emphysema cause difficulties in breathing out? All right, a smooth muscle fiber in the airways are destroyed. Airways is not related to emphysema, because emphysema it's all about the alveolus. So A, wrong. Too little mucus is produced in the bronchi. All right, that doesn't explain. All right, and it's not related to emphysema, is not related to bronchi as well. The elasticity of the alveoli decreases. Yes, elasticity of the alveoli decreases when in emphysema. The surface area of the alveoli becomes larger. No, it actually doesn't become larger, it becomes smaller. So 36C is the correct answer. Question number 37. Which factor contribute to the outbreaks of measles after natural disasters? Contamination of water with untreated sewage. Well, untreated sewage contamination, well, that does not cause measles. So when one is wrong, luckily option A, B, C all cancels out. So D is the correct answer. All right, let's look at the other 2 points. Lack of effective vaccine coverage in the population before the disaster. Yes, definitely. Measles is a pretty hard disease in terms of the vaccination, because multiple booster doses are needed in order to acquire immunization, and also those who have a weak immune system cannot be immunized. People living in overcrowded condition. When people are living in overcrowded condition, so yes, option 3, yes, option 3, overcrowded condition should definitely be considered, because it is one of the main causes for measles to spread. All right, so option 2 and 3 are correct. D is correct. Question number 38. There are two main classes of antibiotics, broad spectrum and narrow spectrum. Broad spectrum antibiotics affect a wide range of bacteria, while narrow spectrum antibiotics affect a specific type of bacteria. Which uses of antibiotics may result in antibiotic resistant bacteria? A broad spectrum antibiotic given to reduce the effect of influenza virus. You see, virus diseases must not use antibiotics, because viruses are not affected by antibiotics. A broad spectrum antibiotic to treat Mycobacterium tuberculosis. We know the specific bacteria that is causing the disease, so we must use a narrow spectrum. Broad spectrum will not be so effective. A narrow spectrum antibiotic to treat diarrhea and sickness in cattle. Yes, while treating animals, antibiotics use can be, all right, can be a problem, all right, that can lead to antibiotic resistance. So one, two, three, all. Option A is correct. People with the autoimmune disease myasthenia gravis can have treatment to relieve the symptoms. In this treatment, one of the components of their blood is removed. Which component is removed? Well, we know that antibodies for the myasthenia, that myasthenia gravis actually causes autoimmune disease, and autoimmune disease is caused by antibodies. So those antibodies should be removed. So option A is the correct answer. 39, option A. Macrophages, neutrophils, red blood cells, they would be unnecessary, to remove them and do treatment with them. So question number 40, why are spleen cells fused with myeloma cells during monoclonal antibodies production? So that the antibodies become specific, for the necessary antigen. Well, no. All right. The spleen cells are fused with myeloma cells so that they can divide indefinitely, so that the resulting hybridoma cells can reproduce indefinitely. Yes, that is the correct answer. Guys, we're done with the question paper. Thank you for joining with me in this particular question paper. If you have any comments, or if you want me to solve any particular question paper for you, please comment in the comment below, and basically subscribe in our channel. We will keep trying to upload every day question papers like this, all right, so that it helps in your exam. Hope this particular video was helpful. Thank you. Bye-bye.